So we're really pleased to have you guys here to listen to and learn from Matt and Faith. Um, Faith has been working with Elaine Ingram at the Soil Food Web, which is one of the, I think, premier places to learn about soil. So I'm really excited to hear what she has to say. And Matt has been working with, is it Clearsun?
Crescive, so Crescive
Cressive.
Soil. Yeah, and they're a husband and wife duo. They've been consultants for about 12 years, and they learned under Dr. Lane 12, 13 years ago. So
Okay, so it's all in the
yeah,
family, really.
same school, but like the original school um, before new ownership came in. So yeah.
Okay. Okay. Yeah, and Matt's specialty is minerals. So
Hmm.
I'm sure we'll hear a little bit about that as we go along. So
Yep.
I'm really pleased to have you guys here. I think soil is something that we just take so for granted and don't really understand the complexity of it. Um, Scientists have been saying that 2/3 of all life on the planet is in the soil. That's an awful lot of critters.
Hmm.
And we don't think about soil as being alive, as being full of critters, as being needed to be taken care of in that way with that kind of understanding. We just see it as stuff that we can move around. And I think shifting from that sort of objective to a more relationship oriented way of looking at soil is actually very enriching, personally and spiritually, as well as much more effective biologically and environmentally. So I'm really excited to see what you have to share. So please share your screen.
Okay.
Um,
You want to do it?
and would you like us to wait until you're done for questions, or are you open to questions while you talk?
Yeah, we can do it while we talk too. Either way is fine. Oops.
Okay, so I'll try and keep track of the chat. If you have a question, either raise your hand or put it in the chat.
Okay, so I'm sorry, I didn't start at the beginning.
Here we go.
Okay. I'm gonna go that way real quick.
Yep.
Okay, so yeah, my name is Faith, and
I'm Matt.
like Catherine was saying, I'm a certified soil food web lab tech, which means I qualify and quantify soil biology with a microscope. And then this fall, I'll finish the program and become a certified consultant.
Yeah, and I'm a water stories student of Zach Weiss. I went and built ponds with him last summer in Oregon. I'm also a student of Crescive Soil, where I've completed courses in mineral balancing.
And then we have a farm, a garlic farm, and then also a business called Soil Dynamics, where we do the soil biology analysis, and then Matt does the mineral recommendations. And today we're going to talk about soil biology and how that relates to plant health. And then at the end, we'll look at some of the experiences we've had on our own farm, especially with dealing with compaction. So to understand soil health, we must first understand the difference between dirt and soil and what's happening on a biological level. So the picture here on the left, this is dirt. And as you can see, there's no structure. It has very fine particles. And if the wind were to blow, the fine particles would be blowing away. And the picture on the right, this is soil. So not only is there a huge color difference, but you can see it has structure and it's holding itself together. So there's no beneficial organisms in dirt. If anything, it's bacteria dominated, which grows weeds very well, or it can have disease-causing organisms. And it's the result of damage to the soil, and this is going to happen through a number of different ways, like applying toxic chemicals, pesticides, inorganic fertilizers, tilling, and heavy equipment. So 3 million tons a year of pesticides are applied globally, which is huge. Herbicides kill weeds and beneficial microbes. Fungicides kill fungi and beneficial microbes. Insecticides kill insects and beneficial microbes. You see where I'm going with this? So anytime you apply these products, it's never just killing its target organism, but also all the beneficials in the soil. And when you don't have any beneficial organisms, there's no competition. So the good guys are not there to outcompete the bad guys. And those disease-causing organisms will overtake the area. Your soil nutrients get tied up in bacteria. You lose your water holding capacity in the soil and also your oxygen space for your roots. And any type of inorganic fertilizer is technically a salt, and putting them down in large quantities is going to kill your beneficial microbes. 80% of soluble inorganic fertilizers are washed out of the soil and into water. In fact, it's so bad in the state of Iowa that the groundwater is not drinkable because it's contaminated with so many nitrates and pesticides. It's become toxic. So tilling. Tilling is going to destroy 50% of your population of soil organisms. And fungi gets hit the hardest because it has these long strands. And then after that, beneficial nematodes and protozoa are impacted. Hardly anyone's going to till just one time. It's either yearly or multiple times throughout the year. And this is especially true with organic growers who can till up to 14 times a year. Now, if you till to a 6-inch depth, you're going to have a compaction layer right at 6 inches. And without beneficial organisms in the soil, you're going to have to continue tilling. It's the only way to plant without the soil recompacting. And disease-causing organisms like root-feeding nematodes, those are the ones that eat the roots of your plant, they live at those compaction layers. And when you plant in the springtime, they come out of those layers because they're attracted to your plant roots, and then they attack it. So roots are not able to move through a compaction layer of more than 150 psi, and only a taproot can move through soil compaction of 300 psi. So this is a penetrometer. It's a really great tool to understand the severity of your compaction issues and at what depth they're occurring. So this picture is showing compaction as getting it at 9 inches and hitting 300 psi. So again, when there's compaction, there's no oxygen, your water can't get in and roots can't go down. So your roots are going to move sideways, and when your water, it's going to go sideways also. When you have roots competing for space and nutrients, that's going to lead to poor plant growth, poor yield, and disease. You lose your soil structure, and your nutrients will actually leach out. Now, if your root zone turns anaerobic from the lack of oxygen, a couple of things are going to happen. The nitrogen in the soil will be lost as ammonia. The phosphorus in the soil will be lost as phosphine gas, and then the sulfur will be lost as hydrogen sulfide. And toxic compounds are made only under anaerobic conditions, especially the production of alcohol. You only need 1 part per million of alcohol to turn your roots to mush, basically, and anaerobic conditions can make 25 parts per million of alcohol. All right, soil. So soil is full of beneficial organisms. There's fungi, bacteria, protozoa, nematodes, microarthropods, worms, and organic matter. These organisms all have special functions within the food web. So a healthy soil will have at least 75,000 different species of bacteria per gram.
Faith, can I ask you a question?
Yes.
About alcohol? Because that's kind of an unusual word to hear with soil.
Yeah.
So what I'm suppositioning from what you said is that it's the anaerobic context that allows for the alcohol to be created. Is that?
Yes, correct.
Okay. And it's a natural thing that happens in soil when it's anaerobic.
Yes.
That's just really interesting because that's just not something I would normally associate.
Yeah, and it's not ideal. You'll have dying plants, so it's not conducive to life. So while you say it, it's natural, it can occur, but it's, a lot of times, most of the times we're worried about alcohol creation. I think when you're making compost piles, that get way too hot and all the oxygen is used up, and the alcohol is what creates the fire.
Oh.
And so that's,
Yeah.
yeah, I don't think in nature, you know, you don't want it to
Right,
occur in nature. So,
right. It's not
yeah.
so likely. Yeah, thank you.
Yeah. And then there's 25,000 different species of fungi per gram. You're going to have over 10,000 protozoa per gram and 25 different species of nematode per gram. And the functions they perform are nutrient cycling, which is the process of making nutrients available to the plants. They suppress disease and pests by competition, inhibition, and consumption. They're going to build soil structure, which increases the water holding capacity, which results in reduced water usage. They help retain your nutrients by preventing runoff and leaching, and suppress weeds, which I'm sure we all could use that. So soil structure is created by bacteria and fungi. Bacteria, they make the smallest building blocks in soil for structure, and they do this by secreting glues to hold together the sand, silt, clays, and organic matter, and they form something called microaggregates. And then fungi are going to grow between these microaggregates and secrete their own glues to bind them together, and they form macroaggregates, so they're really a lot larger. And macroaggregates are what create structure in your soil, allowing space for oxygen around your plant's root zone and space to hold water. In a well-structured soil, as water passes down through the soil, a vacuum is created and it pulls in oxygen, and that creates those aerobic or oxygen conditions for those beneficial microbes. For every 1% increase in soil organic matter, there's a potential for 25,000 gallons of water stored per acre if beneficial biology is present. So to clarify this, if you only add organic matter to your soil, you'll get a little bit better water holding capacity. But to get to the place where the organic matter can reach its fullest water storage potential, you need soil structure, which is created by the beneficial microbes. And this structure allows water to infiltrate down to all the organic matter layers where it gets absorbed and stored by the organic matter. And Dr. Lane actually did some work on a table grape farm in Australia. They had surface level compaction of 300 psi, so nothing was getting through there. The roots were only going down 1 inch, maybe 2 inches at the most. So water would hit that compaction layer at 1 to 2 inches and then go sideways. So they broke through all the compaction and inoculated with beneficial microbes. They were able to reduce their water usage by 50 to 70%. And she has other examples of this in the southwestern United States and Southern California, and found around a 70% reduction in water usage because of that soil structure the microbes created, which increases your water holding capacity. All right, so nutrient cycling. A plant does something very special with its roots. It secretes something called exudates, which is a fancy word for proteins, carbs, and sugars. And it secretes these exudates in very special recipes depending on what it needs. So if it's low in a certain mineral, like calcium, it's going to put out an exudate recipe that's specific to the type of bacteria or fungi that specialize in mining calcium. And also by secreting all of these exudates, it's attracting organisms around its root zone, right where it needs it, creating a protective web of these beneficial microbes. So the first beneficial organisms I'm going to talk about are bacteria and fungi. Bacteria and fungi, they make enzymes, and they use it to pull nutrients out of the sand, silt, clays, and organic matter, and then they store these nutrients inside of their bodies. They become extremely full of these nutrients, but the plant can't access it. It's locked up in them. So they need something called a predator to come along and eat them, and then poop out the excess nutrient. And this turns into a plant available form of that nutrient. And in fact, the nutrient levels are so high in bacteria and fungi that it's toxic to the predators unless they poop out the excess. And this process has a kind of a funny name. It's called the poop loop. So, so over here, this is bacteria. I got a little video. This is under a microscope. And they're these little circles. They kind of move back and forth. They come in all different shapes and sizes. I think the video quality is a little bad. Sorry.
Can you guys see the little
I don't
circles
know
in
if I
there?
can.
Oh yeah.
Yeah.
Okay.
Okay.
And then over here is fungi. They have these long strands.
Yeah, this is a really good thing to see on the microscope.
Yes, we
This
get very excited.
this is what we're looking for. We know we're in a good place, and we're seeing these guys, which are the good guys, versus other things we'll talk about, which are the bad guys. So
All right, so now we'll talk about the predators that eat bacteria and fungi. And the first group is protozoa, and this consists of amoebas and flagellates. So this is an amoeba right here. They are going to eat algae, they eat other flagellates, but they also eat a lot of bacteria. And then over here we have a flagellate. They kind of bumble around. There it is. And these ones only eat bacteria. So once the protozoa eat bacteria, they poop out the excess, and their poop is a plant available form of the nutrients that were mined.
so that's where the soil food web name kind of comes from. Is this food web is predators eating the bacteria and fungi in that whole cycle. So
And then the next group is nematodes. So these are named based on their function. Bacteria feeding nematodes eat bacteria. Fungal feeding nematodes eat fungi. And predatory nematodes eat other nematodes. And they're part of this poop loop as well. And nematodes actually create pore space, which allows for more water storage and air in your soil. And most people have probably heard of root-feeding nematodes, which attack the roots of your plants. The cool thing is that these beneficial nematodes, like the ones I listed, will inhibit these root-feeding nematodes from getting to your plant. And predatory nematodes actually really love to eat root-feeding nematodes. They will go after those ones first. So there's other predators like microarthropods and worms, and it kind of gets higher and higher. But without these predators, nutrients are locked in the soil by fungi and bacteria. So they're very vital to having a healthy and a functioning soil food web here. Got a little video of a nematode moving around. They're fun to watch.
should we talk about the now?
It's amazing how active they can be in dirt.
Oh, yes. Yeah.
Yeah.
All right, so now we'll talk about anaerobic organisms. So these are organisms that thrive in no oxygen conditions. They do not make glues or form aggregates of any kind, and they're not performing nutrient cycling. So you can find these organisms in areas of compaction with overwatering events like flooding or just even irrigating incorrectly, in compost that was not made correctly. And what we see a lot of is when someone makes a compost tea and they add too much food, which is really common for people to do. The organisms in that tea reproduce so fast that they use up all the oxygen, and then it turns anaerobic. And then this is spread out onto a field or garden, which spreads the organisms further. And real quick, I want to say thank you to Sarah from Crescent Soil. She shared her anaerobic organism pictures with me. I don't have a big library of these, so just want to tell her thank you for that. All right, anaerobic bacteria. So Spirilla, spirochetes, and Vibrio are anaerobic bacteria in the soil. And when you see these organisms, you have to take it very seriously, as these are the conditions that human pathogens live in and can't be ruled out. So you'd get very sick if you touched soil that had these in them and you touched your face. This is a video of a spirochete.
Kind of see him wiggling back here.
Yeah, it's kind of hard to see.
Yeah, kind of just flopping around. It's not a good one.
All right, oomycetes. So this is a category for disease-causing fungi. And they are not building soil, they're not performing nutrient cycling, but rather their function is to consume living plant material. So we don't want to see these.
Yep.
Yeah.
Bad guys.
All right, facultative anaerobes. They live in the middle between aerobic and anaerobic. And this is a video of a type of protozoa called a ciliate. And we start to see them when the oxygen levels drop. And it's a good indicator that our soil is starting to go anaerobic. And it's something we see a lot when someone does a lot of overwatering. Actinobacteria. This is a type of bacteria that hovers in that in-between zone as well. And if it's high in numbers, that's an indicator that things are going anaerobic. Certain types of plants need actinobacteria, such as brassicas, and that's because they inhibit the growth of mycorrhizal fungi, which is detrimental to brassicas.
And it's these little thin strands here you're seeing.
So there are many common practices that appear to be helpful, but end up destroying our beneficial microbes. I'm going to go over the few of the most common ones that are practiced. So buying and applying compost seems like a great idea. However, depending where you get your compost from, you could be adding heavy metals and toxins and next to no biology except for bacteria, which ties up your nutrients. Many home gardeners will make their own compost and apply it to their garden also. But there's many nuances with composting, which include correct temperature targets for the right amount of time, moisture management, and ratio of ingredients that it can be difficult to get right. And if these are not met correctly, then the causing organisms will overtake the pile. I realize it's not practical for everyone to use a microscope, so there are some good indicators that clue you in on what's going on. So for these anaerobic piles, the bad ones we don't want to have, they're going to be black in color instead of a nice dark brown. And they will stink. They will smell like ammonia or rotten eggs. And these contain very toxic compounds. And like I said before, that anaerobic conditions produce alcohols. So if this pile heats up to 180 degrees Fahrenheit, it will burst into flames, and this is called an alcohol fire. And if you are buying compost and they tell you not to plant directly in the compost because they'll kill your plant, that's not good compost. Or if the compost is still hot to touch and hasn't cooled down from the composting process, it hasn't finished composting. So that's also not something you want to put in your soil right away. Now, aerobic piles, so the good ones, they're going to be a nice dark brown color. They smell good. They will have formed aggregates. And you can plant directly into it and your plant will do very well. These piles will have reached the high temperatures required to kill human pathogens, pests, plant pathogens, and seeds. So if you put seeds in your compost pile and you finish that whole composting process and those seeds start to sprout, then you know your pile never got hot enough to have killed those seeds.
So is temperature something that you can use to sort of see things going anaerobic?
Yes. So yeah, if you get up into, once you hit 180, you know you went anaerobic. I mean, we want to turn a pile before it even gets close to that to prevent it from happening.
What would be the temperature at which you'd want to turn?
It depends. So depending what you use for a high nitrogen source, it will go really high fast or it'll take a little longer. You want to hit 131 for a certain amount of time. And then there's the next category. If you get even higher, it's only going to be for 2 days. Or if it hits 172, it's only, 12 hours. So it's on a scale.
Yeah. I think the school is teaching if it hits 170, turn right now. It's too high. So if it hits over 170, even though you might have a little wiggle room, I think they're teaching 170 now.
Okay.
All right, so organic pest control products. Neem oil is probably the most popular, and it's commonly used among home growers and organic producers because it's sold organic and people feel safe using it because it's more natural and comes from a plant. However, due to the way neem oil is processed, it actually becomes contaminated with many toxic pesticides. This is a report from the Oregon Department of Agriculture. They had a grower get his crop rejected because it was contaminated with synthetic pesticides, but the grower only used organic pesticides. So the department looked into what he was using and got it tested. They checked 6 nationwide brands of neem oil being used for organic crops. Every single one was contaminated. And this is the report. It's showing 9 pesticides found in neem oil. So once you apply this to your soil, you've now contaminated your soil with pesticides. And neem oil is also extremely antimicrobial. One light application can reduce the soil biology populations by 84%. And water management is another one. So getting the watering just right is something that's commonly overlooked. Overwatering will create disease-causing organisms, and underwatering creates conditions where your biology can't survive. And most people go through what we call a wet-dry cycle, and this is where you water deeply and get your soil nice and wet. However, many growers do not maintain a consistent moisture level in their soil, and the soil dries out until the next watering. And then this cycle repeats. So it gets wet and then it dries out. Well, your biology is going to struggle with these repeated cycles. And the cycle is especially hard on the soil predators, which are needed to perform the nutrient cycling. They'll either go dormant or they'll die. So getting the watering right is challenging, but it's something we should all look out for to help our microbes out. All right, so what you can do to have healthy soil. When hot sun is on bare soil, it's going to bake it and dry it out, which affects your biology very negatively. Temperatures can be 40 degrees hotter on bare soil than on soil that's covered. Also, when rain hits bare soil, it will cause rapid compaction, and that will result in the water staying above that compacted layer and then running off. And mulching is important because it insulates and protects the biology, and it helps prevent moisture loss. A mulch layer is also a really good source of food for the microbes. And we like to use mulches that feed the fungi, like wood chips and straw, but it is difficult to find clean straw that's not been sprayed. All right, so Matt's going to take over from here.
All right. Give Faith's voice a little break here. So yeah, dealing with compaction is really essential for having healthy soil. If roots and water can't penetrate, then you're not going to have a healthy crop, and disease can occur. So when you think about the depth that roots should be able to get to, it really opens your mind up to how detrimental compaction can really be. So how deep do you think ryegrass roots will go without compaction? So try to think of a number in your head. Ryegrass, how deep can those roots really go? So they can actually go down to 15 feet without compaction. And so blueberry plants, I know Dr. Lane was talking about this one on a podcast, but blueberry plants, you think about how deep can those roots go? She was saying 25 feet with the right biology and in some of her practices that she does. So that's pretty impressive, I think. And it's getting all the way down there and getting the water and the nutrients at those levels as well. And so it's just going to be a much more resilient plant if you can get roots that even come close to that far down. So there's multiple ways to deal with compaction. You really have to pick the management method that's appropriate for your context. So like for small scale backyard gardens, using a broad fork is a great option. Adding the soil biology immediately after broad forking will inoculate the ground and further break up that compaction and start the process of building that structure that we were talking about earlier, which is the sponge. If you heard the term soil sponge, it's this biology that's creating structure that's soaking up the water while also pulling down and sucking down oxygen. So tilling is another option, a management method for compaction. You should also, again, inoculate the ground with biology when you till. And the important point here is this should be the last time that you'll need to till. So the idea here is you want to break up that compaction one last time. This is what Dr. Lane does teach. If we just apply biology just straight onto compacted soil, it can take, I think I have this later in the notes, but it's like 20 or 30 years if you don't break it up first versus breaking it up and then getting the biology in there with the extract. So, and then for larger operations, a deep ripper works or a yeoman's plow, that's another thing Dr. Lane really teaches, is that yeoman's plow is great for basically, without disturbing the topsoil too much, getting down and ripping that subsoil that's highly compacted, and then coming along with the drip extract right behind that. So they'll actually have these extract the equipment set up to drip right as the plow goes through. It's dripping extract as it goes along, which is pretty neat. So yeah, to inoculate the soil, we prefer compost extract. It's the act of squeezing compost through a mesh bag into water. So you massage the compost for up to 5 minutes to extract the organisms out of the compost. It's more practical and affordable than applying compost in its solid form. So to just extract it into water and spraying that out, you're going to get a lot more use out of that compost than just applying compost in its solid form, especially when you have really good compost that costs a lot of money because it's been verified with a microscope and someone from the Soil Food Web School. Yeah, again, you can dilute the extract and spread it out over your property, garden, or farm. It usually takes multiple applications over time to inoculate your soil with the biological levels that you really need to do all these great things that we talked about, that Faith was talking about with building structure and nutrient cycling. So if you were to only use compost extract, again, it'll take you 20 to 30 years without breaking up that compaction first. And that's a big point. After breaking up compaction, inoculating via that extract is the only way to get things moving in a reasonable amount of time. Bottom line is what we're getting at there. All right, so mineral applications. Most soils, and especially in an agricultural setting, we're gonna kind of talk a little more kind of an ag setting somewhat, but it's still applicable to really any plants on any scale, whether you're a back home gardener, whatever it is. So most soils in an ag setting are out of balance and deficient with minerals, especially if you're planting the same crops in the soil, time after time. And it's going to draw a lot of those minerals out of the soil. So applying a traditional NPK fertilizer or any other type of nutrient can further send the minerals out of balance if you don't understand your current levels of nutrients with lab testing. So how can you know what you need to put into your soil from a mineral context if you don't actually know what's already there and what's missing? So we really like to push the lab testing to see what's there and then balance the minerals from there. So you could be applying too much potassium, magnesium, sulfur, etc., and further put your soil out of balance to problematic levels if you're not actually using the testing and balancing accordingly. So an example of this is if you have too much potassium in your soil and not enough calcium, plants are forced to use potassium to build their cell walls, which makes them weak and watery and susceptible to disease. It's very common in traditional ag. They're really pushing a lot of potassium products because it gets a lot more profits. I think I'll leave it at that. But what happens is you get a weak plant, and you've got to use other things and purchase other things to try to fix that issue. So if the calcium and magnesium ratio is out of balance, the soil will be tight and compacted. It's especially important in clay soils. So I'm not going to get too far into the mineral balancing. It probably would require its own webinar to fully understand everything, but it's vital for healthy plants and nutrient-dense foods for sure. So organic matter. So we need at least 3% organic matter in our soils for microbes to actually stick around. To add organic matter to your soil, you can use a mix of different cover crops, mulches. You can grow a permanent low-growing ground cover, such as creeping thyme, creeping speedwell, white clover. Those are things that the Soil Food Web School and Dr. Lane teach quite a bit, is to have a perennial low-growing ground cover that will keep that biology moving and keep it active and alive in your soil. So you can also use compost mixed into the soil if you have enough of it, depending on your scale. But so organic matter is a food source for the microbes. They need the organic matter to survive and thrive. We stay away from the organic matter sources that microbes are not able to break down. So that's really important. We started.
Could you say more about what organic matter would they not be able to break down?
Well, there's still a lot of research to be done on this, but biochar is an example of something that a lot of the soil food web and some of the colleagues we've had in this community using the microscope to quantify and qualify the biology. Biochar is something they're finding is it's really hard for microbes to break down. So it's such a stable form of carbon that doesn't readily break down from the microbes. You're really better off as far as if we're just talking about organic matter and a food source for the microbes, I'm just talking in that context specifically,
Okay.
then you're better off using wood chips versus a biochar, or again, those other things we talked about, other mulches, they're just going to break down more naturally and quickly and be a better food source for these microbes that we're talking about.
Okay.
So.
Is there anything else other than biochar that might be difficult?
That's kind of the big one, because it's such a big thing right now, especially. And so that's one that's getting talked about quite a bit. And so generally the organic matter, again, that breaks down more readily are those other sources, and you're better off as far as food for microbes, just using those other ones. So I can't think of anything else. No, just the biochar is kind of the main one here. So yeah, we're just going to talk about our farm for a little bit here and our experiences to pass along. But we started our garlic farm over 3 years ago, and starting at year 2, we began the process of inoculating our soil with the biology. So we discovered the soil food web approach at that time. We wanted to find a way to grow organically and without traditional fertilizers and pest management practices. So we noticed in our home garden that even growing organically still led to pest and yield issues. And after a year farming garlic, we discovered again the soil food web. Our second year of growing was when we used biology applications only, and we saw a really good yield increase and minimal disease in our crop. Our third year, however, was a completely different story. So in the third year, we discovered that even with great biological numbers and nutrient cycling, we had smaller bulbs and still noticed disease in a portion of the garlic bulbs. We had a 6.5 increase in yield our second year, but that dropped to the 4.5 times increase in year 3. And what we mean by that is basically we plant the clove. That first year was 6.5 times what we planted, so that clove grew to approximately 6.5 times, versus in the year after that, in year 3, it only grew 4.5 times. And so this is when we jumped into mineral balancing, because we discovered, luckily we came across Crescent Soil who's lived it and has done it for 12 years and has been flown around the world, similar to Dr. Lane, doing consultant work. So what they discovered is, especially with hungry plants, the biology helps that plant really draw down the minerals big time. And we're talking, again, agricultural setting here, kind of high production greenhouses, and in market garden situations where it's high turnover, you're putting crops in there constantly, and trying to get the biggest best yield that you can, and having high biology, it really draws down those minerals. So we found Scott and Sarah at Crescent Soil. We talked about them a little bit already. They went through the Soil Food Web program about 13 years ago. They've been very successful as consultants because they included the mineral balancing along with the biology. So again, we're talking kind of small scale growing here, where it's financially practical to but purchase all of these different minerals and trace minerals and all of these different things to apply, on a smaller scale. Luckily for us, they had classes on mineral balancing, which I took, and I learned by using a Mehlich 3 test that we were able to identify our deficiencies and increase our minerals to our plants with the help of the microbes. So we will have our data for results this fall. So excited to share that later on this year. All right, so soil biology results. We did soil assessments of biology when we planted the garlic in the fall and then compared it to what it looks like now. So we increased our fungi by 177%, kept bacteria in a good range. Again, you don't want bacteria to get too crazy because that can mean, especially in the presence of very little predators, that the bacteria really high will lock up those nutrients if you don't have the predators in the full soil food web to consume the bacteria and release all those nutrients. So yeah, we increased by 177% of the fungi, good protozoan numbers, and again, that bacteria was in a good range. So we encountered issues with our own water management. We were watering deeply every 3 days, but this led to a wet-dry cycle that made our protozoan numbers actually go down. So it hurt our predators, like Faith was talking about earlier. So after we changed our water patterns, we were able to increase those protozoan numbers and keep things at more of a steady state.
How did you change? What was the difference?
It's essentially just watering more frequently and like a less amount, because you don't want to drench your soil and create those conditions that Faith was talking about, but you want it to be pretty consistent moisture level if you can, even if that doesn't necessarily happen in nature. I'm kind of talking like a, again, high production, market garden kind of agricultural setting. If you can help it, it's best for everything, your plant, the microbes, the soil in general. If you can kind of keep that moisture in a steady state versus these high peaks and troughs of your water levels and your moisture levels in the soil, rather. So
did you find you were using less water by doing that or more?
I think it was roughly the same, kind of the way we did it. It was roughly the same. If anything, maybe a little bit less. We're still fine tuning it. We're only a few years into this, and it's kind of a lifelong experiment.
Yeah. And a year is,
So,
you have to have a year, to see
Yeah.
what it's going to do. It's just,
yeah, right. So there's, our crops like ours, we get one shot. It's a 9-month growth cycle. And so you get one shot to do it. And so we have, just every year we're getting data for that year. There's other crops where you can have, in a greenhouse setting, you can have like four harvest cycles. And if you're tracking the data in 4 harvest cycles, that gets you data 4 times as fast to fine tune things. You're going to learn much faster in a setting like that, depending on the crop. So yeah, that's some of the numbers, soil biology numbers, and then we're just gonna walk through a little bit on how we attempted to address a severely compacted 1/4 acre and discuss the mistake we made, basically, and what we've learned from it as far as compaction. So we grew our farm from a 1/4 acre to a 1/2 an acre last year, and we faced serious compaction issues in the new zone that we were creating. There was compaction at 1 to 2 inches depth of 300 psi. So, nothing's getting through there. Again, 150 psi is the max of what a plant's roots will grow through. So at that point, wherever it reaches 150 psi, the roots are going sideways. The water, I think, is going sideways. The water might be able to get through a little bit more, but a lot of it's going to be going sideways at least. So this area that we're talking about was run over by a tractor constantly the year before, and it always had standing water after a rain event. And so essentially only weeds were growing from it. We did not have the ability to use a deep ripper or yeoman's plow at the time. We're actually getting our hands on one through a family connection later this year, which we're excited about, to actually break up that hard pan layer. So in order to break up the compaction, we started with a broad fork. As you can see in these pictures, the large rock looking chunks, they're not rocks, it's actually soil that we had to smack with the broad fork in order to break it up. We tried to use a tiller at first, but it was struggling to get very deep. It was kind of bouncing off of this layer of compaction, and so even the tiller couldn't even get into it. And so, yeah, you know, not a good outlook, but we're slowly kind of getting through it, as we'll talk about, and you can turn around, you know, pretty horrible soils. I think it's kind of the hopeful message that we're going to talk about here. As horrible as this soil sounds. So next we applied a compost extract to the broad fork area, followed by our mineral applications. It was tilled into the soil to a 6-inch depth, so you can see the minerals here, and that's me with the tiller tilling those minerals in, as well as the biology, the extracts that we were doing. We were tilling that in as well, and we were doing this with the minerals so that we can apply more minerals at once, because we essentially, like I said, we get one shot to apply a bunch of minerals. And if you can till it into 6 inches, then you can apply a little bit more. You can calculate to 6 inches without getting into too much, instead of just applying enough for 4 inches. So yeah, so we did that. We added another compost extract to top it off, and then covered the soil with wood chips until it was time to plant, in order to protect the biology and the moisture levels. Always want to mulch if we can. And we prefer wood chips because the wood chips is actually a food for the fungi. So the fungi can break down mulches that are high carbon. And so you're going to be feeding the fungi. And that's always what we're after to feed in the soil, is the fungi. Bacteria don't have a problem. You know, you very rarely ever have to add foods for bacteria to come back, unless there's been so many protozoan and predator numbers that they've collapsed your bacterial numbers so much. So we're really after feeding that fungi, creating those macro aggregates that Faith talked about, and really creating that soil sponge. So really,
Where do you get your, I'm sorry.
oh no, yeah, where do we get our wood chips?
Yeah, there you go.
So we have 5 acres, and so we've chipped up a lot, just dead trees, mostly on our own farm, but a lot of it has come from Chip Drop, and so we recommend Chip Drop. If you guys haven't heard of it, yeah, it's just chipdrop.com, I think, or getchips.com or getwoodchips.com maybe, but it's called Chip Drop. So we recommend that. And the arborists, you'll just get on a map that the arborists will use to see if you're close to a job they've done. They'll choose your place and come drop chips. You can go down there for free. You can donate. You can basically put on there that you'll give them $20. You'll give them your card number on the Chip Drop site, and then when they drop your chips, they'll just charge you $20. Which, for a full load of wood chips, I'm okay paying that. So I will say, as far as wood chips and in the biology, the chips, and especially if you're going to be composting with wood chips, which I think everyone should, because it feeds the fungi once again. But something to note is certain species of trees, like pine, so fresh pine has pinic acid, which will inhibit the microbial growth, and so it's not going to be as beneficial. You'll want to age it for like a year before you try to compost with it. And so yeah, something to think about. There's certain species. Cedar obviously is naturally antimicrobial. So some things to think about with the different species. So
Lori has a question too. She said, is your soil, I'm not going to say this right, caliche, calcium carbonate? Lori, do you want to pipe in here?
We've added calcium carbonate in the form of like oyster shell, we've had to put a lot of that in our soil because we were so high in potassium. It was out of balance with calcium.
Yeah, it just looked when you showed the picture of it, it looked those clumps that did it have a kind of whitish color to it?
Oh,
I'm
the compacted
so
one?
Oh, right here.
yeah, yeah, the compacted one. So I'm in Texas and we have caliche and it's the high calcium carbonate.
Okay, gotcha.
And so it feels like you're basically gardening in rock.
Mm.
And
Yeah.
it'll soften with rain, but you really, I mean, sometimes you have to use a jackhammer to get through it.
Oh, wow.
Wow.
Yeah. Yeah,
So
ours has softened up a little bit. I didn't know it was called that, calcium carbonate. I think when I hear calcium carbonate, sometimes that's an actual amendment you can put in your soil and balance things out with the magnesium and potassium. But yeah, so maybe that we probably have the same thing you do. I don't know.
it's like it seems like you're working with limestone sometimes. Is that it? And in Texas, I mean, well, we're known to have really high pH, like up to 8.5.
No, we don't have that.
No, we
Yeah, I
don't. We
don't have
have
that.
it's like a 6.4 is actually really nice
Oh,
pH, like a
wow.
natural pH. And kind of what a lot of plants like to have. And so we haven't seen that. I think we have a lot of basalt where we are at. We had a lot of decomposed granite
Okay.
when we built our shop. A lot of that was getting shot out of the ground from this huge auger that was making holes for our posts for our pole barn. We
Mm,
have a lot of decomposed granite. But yeah, and then basalt is everywhere, it seems like, as well. But
mhm.
yeah, so I don't think we have it quite as bad as you have down there,
Yeah,
limestone.
yeah, yeah, definitely. It sounds like you're working with a different composition for
Yeah.
sure.
Yeah,
Yeah. Okay. Thank you.
yeah. Let's see. I think we're basically here at this compaction slide. So we learned firsthand that compaction is hard to undo. And even with the tilling and inoculation, we didn't get deep enough for our crop in some areas. So we used what we had at the time, made the best of it with the broad fork. Again, this fall, we plan to use the deep ripper we have access to, which is great. And really get through that hard pan. And Scott, my one of my teachers through Cressive, was talking about using like a trash pump if you have it, and essentially after you break through that hard pan with the deep ripper, if you have a trash pump hooked up to, you can use like an IBC tote if you have those, or you can get your hands on one, and it's basically got a connection at the bottom. If you can do an extract in a trash pump and spray it out all over your fields after you break up that compaction, again, we're inoculating with that compost extract to get the structure starting to build underneath after you've fractured that hard pan. Getting that biology to do its work and building that soil sponge, is really important. And that's one way you can do it. You can use a sump pump, a submersible pump. I'm going onto a tangent with equipment a little bit here, but there's ways out there to look up how to do these. If you Google it, that's a really good one with the sump pump because it's not too hard on the biology. And so just a garden hose and a sump pump, submersible pump, you can pump out that compost extract and again get that structure building and under your soil there. So overall, if we had to do it again, we would have started with the deep ripping. We learned a valuable lesson about compaction and how we needed a way to get deeper than the tiller and the broad fork allowed for our particular farm size. So
I'd like to ask a quick question about compost extract. You had said in earlier that you put it in a bag and squeeze it. And I think for maybe a home gardener, that might not be too big a deal. But you're doing a larger area. Is there a technique you use to do that to get a larger batch of
we
extract?
we
Or
just do more bags.
Yeah.
So
okay,
we have two 55-gallon drums that we fill with warm water. So if you can do a tepid kind of a warmer water, it's kind of like you and I, we want a warm bath. We don't want a cold bath, and the microbes are kind of the same. So you'll make them happier and they won't go dormant if you are using the warm water. And we'll just do it in 2 55-gallon drums, and then I've got a tractor that'll pick them up because they're sitting in a an IBC tote with the tub pulled out, and so you just have the cage, and then I can lift that up with the tractor forks and go set it in the middle of the farm. And then it's the setup I just talked about with the sump pump and garden hoses as what we use to dispense that compost extract. The bag that you want to use is 400 microns is what you're shooting for these mesh bags. So you don't want it any tighter of a mesh than that, because the biology is not going to be able to get through that mesh bag. So 400 micron mesh bag is what you're looking for to do those compost extracts. And yeah, anybody can buy good compost and just extract it themselves using a 400 micron mesh bag, just in a 5-gallon bucket. And
If you don't have a mesh bag though, you could squeeze it with your hands in there and just try to squeeze the compost that way too. So
I had pictures of stomping grapes. Yeah.
yeah, but I can't emphasize enough the importance of buying good compost that's been verified, ideally by someone through the Soil Food Web. So the Catalyst Bioamendments recommend their compost, or we actually prefer, the best stuff is to us is Cressive's Cressive soil. They're
not paying us to say that.
No, they're not. No, not at all. Well, we've gotten both, and that's what we prefer. So
What about worm castings?
Those can be good, but the same thing, it depends on what the worms were fed. If they were fed a lot of green foods, that's feeding all that bacteria, that's not good. Or if they're feeding more complex carbs. And then you kind of have to just check it under a microscope to know for sure what's going on there. And they have to stay at a 50% moisture, I believe. So if you have dried worm castings, probably all that biology is not in there anymore.
yeah, we've actually bought some worm castings, and we checked it out under the scope, and there wasn't a whole lot
There
going
wasn't
on.
anything there, but they were completely dry. So
Yeah,
Yeah.
in comparison to really good compost made through with Soil Food Web principles and methods rather, that was the best stuff you could put on your
well, a lot of
plants.
people put worms in their compost too, just further makes it even better. So
yeah, because you can even take a pile that's just not really great, and you add worms to it, and they can make it,
just give it some time.
give it some time. Yeah, it takes longer that way, but it's yeah, it'll make a really, really good compost pile. So in conclusion, so healthy soil can take time to build and repair, especially in degraded landscapes. But the benefits in the long run of using biology are well worth it, as it results again in water retention and healthier plants. So building that soil structure and getting that nutrient cycling happening in your soils, that's what the Soil Food Web really is all about, and good soil biology is all about. So, and that's all we had.
Yeah.
Well, and I want to say a little bit about Elaine Ingram and the Soil Food Web School, because we're so used to doing compost, and we just assume that if it's made at home, it's got to be good, right?
Right.
But it is what I have learned on this journey is that it just is not so, and there's no way to tell. I mean, it all looks pretty good. If it's brown, you assume it's fine. It's only with the microscope that you can really see these things, and I think that's and to my knowledge, she's the only one who's really pushing this level of scientific awareness.
Yeah.
And graduates are trained to use a microscope, trained to really know what to look for, how to count, what they're looking for when they're counting, all of that. So it may seem to folks who aren't used to it that it's like a bit much. But I do think because the living things in soil are so tiny You really need to have a way of looking at them that gives you real time data. You're not guessing. You really can actually see what it is. So I think it's really worth it. So I'm just really thrilled to have you guys as part of Soil Smart. And we're really encouraging people to take advantage of your skills, because knowing what's in your soil, knowing what's in the compost you make, really helps you figure out what you need to do to get the results that you want. I think it's taking composting to a different level. The other thing that Faith mentioned, and we did have Nick Tomasini talked about it a few weeks ago, is temperature, and how important monitoring temperature is to really get the kind of compost that you want, especially if it's microfungi that you're hoping to have at the end, which again is sort of a new concept for people in terms of compost, in my view. You don't talk about that much. You just assume it somehow magically happens, instead of understanding you can create the conditions for it if you pay attention. So we'll be doing more in those areas and helping people be able to figure out their own process for making that happen in their areas as we go. So thanks so much for you guys. Are there any questions that people have before I?
Yes, I have a question.
Okay.
Could you say a little bit more or give a brief description of the mineral balancing that you were talking about? Is it based on, I mean, some of the Albrecht's ratio?
Yeah.
Oh,
Yeah.
you know him?
Yeah, you know Albrecht.
Yeah,
Okay.
yeah.
Yep, absolutely. And so the teacher, again, the school that we went through for mineral balancing was the Crescent Soil School, and it's based off of the Albrecht method using the Mehlich 3 tests, and
Okay.
really focusing on balancing first before raising your levels. And so you're balancing the cations at first. And so the Scott and Sarah from Crescent always say balance before volume. And so what you're doing is you're balancing calcium, magnesium, potassium. Or those are the kind of the primary ones. Sodium's included as a cation, but you generally just want to keep sodium low for the most part. And so you're balancing these major cations. And what he discovered is so his story, and it just in case anybody hasn't heard of him, and you know what his story was all about. Essentially, he made some discoveries after the draft in World War II. He was wondering why there were so many draftees that were being rejected from certain areas, and more draftees were accepted in other areas of the country, primarily the Midwest and some of those regions in the Midwest. That there's more draftees going into World War II from those areas versus like in Appalachia. There were more of the draftees that were rejected for the draft, and he was wondering why. And he came up with the research that essentially proved that the soil was the reason why, and its health, how it related to human health. So he discovered that the soils were just in better balance and richer in the Midwest versus Appalachia. And so he spun down soil. He did a lot of research, and he spun down soil for like weeks on end to separate it into its different parts, and found that the ideal soil, there's a book you can actually look into, probably the #1 book that you would want to check out as far as the Albrecht method is The Ideal Soil. And he gives you all the numbers in there of what the ideal soil is, mineral balanced soil. And it's like 68% calcium versus 5% potassium. And then essentially, I think 12 to 14% of the magnesium is in there, is what you're looking for. And that's called the base saturation. So without getting into it too much, yeah, I guess to sum up the Albrecht story, so he made these discoveries. He was working for the University of Missouri at the time, who was taking a lot of grant money from certain corporations, and he was really making healthy plants with some of his discovery, and it was not good for profits. And so he was let go. And so it was kind of a sad story, but his research really lived on, and farmers are some farmers are using it and to great success, including our own farm. So he still had a big following and wrote books and had success. So but yeah, that's kind of his story and the power of corporations. So anyway, yeah, I kind of went on a tangent there, but
but very fascinating. Thank you so much.
yeah. But the ideal soil, I would check that out if you want to learn more about his method and how to balance your own soil. But yeah, there's more minerals than that. Obviously, there's trace minerals that you want to raise and get your levels up to as well. So sulfur, phosphorus, others than just those major cations I talked about. But yeah.
Any other questions? So I want to say a little bit here, because Soil Smart is really focused on the biotic pump. And for those of you who don't know what that is, it is how nature cools the planet. It's how we can get out of this mess we're in, actually, because we're getting warmer. And so the balance to warmer is cooler. And the thing that makes things cooler is water. But as you may have noticed, we have a lot of water in the air, but we don't have as much water in the soil. And in fact, we have lost 2,623 gigatons of water in the last 17 years worldwide from soils. A gigaton is 1 billion tons. That's an awful lot of fresh water, actually, that has disappeared. And as the land gets dry, as they have said already, water, when it falls, doesn't stay. And we're seeing that. It rushes off in huge, huge torrents. So it's really important that the soil be healthy, not just for food. It's great to have it for food. It's great to have it for your plants. But every piece of soil on your property needs to be spongy to hold water. You don't want to have any water run off your land when it rains. That's the goal. That's our goal for the city of Spokane, actually, is to make Spokane a sponge city that will keep the rain water it gets. We get 17 billion gallons of water a year at 17 inches, which is very dry. But if we kept that water in the soil, we would not need to use so much water from the aquifers. We would be able to preserve the beautiful trees that we have without a lot of additional watering. It would be much cooler, because soil temperatures, as Faye said, are way cooler when the soil is damp or moist or holding water. So it's really, for us at Soil Smart, a very wonderful approach to climate change. I think it really engages us in a relationship with the planet as nothing else does. So I encourage you to become a member of Soil Smart. You've signed up tonight for free webinars, and that's fantastic. If you keep the URL, you can come back and revisit this at any time. You have what is a limited membership, which means you can see what you signed up for, and you can sign up for anything that's free. So you can sign up for all those free webinars and you can go see them all. However, if you're a member, you don't have to sign up. You just get to see them, and you can see them at any time, and you don't have to keep the URLs. And we're doing other things as we grow. So we'll be doing other kinds of classes. I'm really hoping to do composting. We're doing some land listening, really understanding how water and sun move across a piece of property so that you can help it stay. So a spongy soil is part of that. But the other part is that water takes time to go down through soil. It doesn't happen overnight. So it has to have places to rest, to spread out, to slow and sink. And that's land design. So this is something that Matt's been trained in a little bit, and we'll be developing more as we go on, so that we really can offer that as a service, so you can get an understanding of your own property and be able to manage it better. We would love to have you donate. So far, we're totally volunteer run. It would be really nice to change that. So you can donate to us as an organization. You can donate to forests. One of the things that we're doing is helping build mini forests using the Miyawaki method. And we're supporting a young man who's got a project at Whitworth in doing this. So the biotic pump is composed of plants, which are the pump part. They pump water from the soil if the soil is healthy enough to hold it, and they evaporate it right into the air, which we're all told about, and that goes into the clouds. But they also evaporate at the same time bacteria, and that bacteria is what allows the water to coalesce around itself and fall as rain. And it falls at lower temperatures when that water collates around bacteria than it does around dust or silver or other metals that might be in the air. So it's really important to allow that circulation of water. Is soil
all around the world, traditional wisdom.
healthy? Go ahead.
Sorry.
I'm sorry, I was looking up a link just on the biotic pump. I'll put in the chat. So it'll
Okay.
lead you and it started playing. Didn't mean for that. Sorry about that.
And we have a whole page on our website also. So if the soil is healthy and can hold it, and then is designed to allow water to sit long enough to sink, and then the plants pull it up, we are much more likely to have rain. So we can create the climate we want if we do it correctly. And that is a much cooler climate than we will have other ways. So you can donate to that, or as we said, we really want to encourage a deeper understanding of soil and land use. So we are offering scholarships, and you can donate to those scholarships as well. So we want to train more people like Matt and Faith, so that we have more resources here locally to do that. So you can do that as well. Any other questions for folks? Really appreciate your being here. Hope we see you back. Next week, this month, we're having 3 webinars in a row. So next week is Hart Hagen, and he's going to be talking about dry soil and that crisis worldwide, which nobody's really talking about. And it is a huge crisis, And then the week after that, we will have Mitch Rollick, who is going to showcase a tool that he's put together to help people understand how water and sun move across the land. So you can actually see in 3Ds and color what your piece of land looks like. So it can help you manage that for yourself. So thank you, everybody, for coming. Really appreciate it. And hope to see you next week.
Thank you very much.
Thanks, Catherine.
Very useful.
Very good.
Thank you all.
Thank you
Bye.
both.
Bye.
Bye-bye.